During last class, we took a look at DNA and the structure of DNA. So DNA is an example of a macromolecule called nucleic acid, and a nucleic acid is made up of many nucleotides. So here's a picture of one nucleotide, and if you can see, there's three different parts to the nucleotide. So I want you to name each of these structures, structure A, B, and C. Structure A is a sugar, and that sugar has a special name for DNA. It's called deoxyribose. Structure B, this rectangle, is a base. We call it a nitrogen base, and there's four types of bases. We have adenine, thymine, cytosine, and guanine. And then structure C, the circle, is a phosphate group. We can take this one nucleotide and put them with other nucleotides to make the structure of DNA. So this is part of your homework where you had to take a look at this picture of the DNA and you had to circle one nucleotide, label a sugar, a phosphate, and then fill in the green boxes with the missing nitrogen bases. You needed to circle one nucleotide, and one nucleotide, remember, is made up of those three parts: the phosphate group, a sugar called deoxyribose, and then a base. Then you had to label one sugar. Now there's many sugars here, and the sugars are those pentagons, so you could have just labeled one of them. And the phosphates are any of these circles that are on the picture. And then the last part is you had to fill in these green boxes, because if one side of the DNA had these bases ATG, CAC, what would be the other side? What would be the matching, or we call it complementary, bases that would be on the other side? Now remember, adenine, A, always matches with thymine, T. So here I see T for thymine, so that means I need A for adenine to match it, okay? So you can think all of the bases that have straight lines, they match with each other. So A and T always match with each other. Okay, here we have G, guanine. It matches with the base cytosine, so that also means that cytosine should match with guanine. So here the bases that are curved, that have curved lines, match with each other. So G to C and C to G. Here we have adenine. What should it match with? T for thymine. And the last one is C for cytosine. It should match with G for guanine. On last class's work, there was also a section where it talked about how DNA replicates or copies itself. Because remember, when our cells need to make more of themselves, we need to make sure we have copies of the DNA. So this is how it works. The DNA, here's the DNA, and here's another picture of it. And what happens is it splits up so that one side of the DNA is split up from the other side of the DNA. So the DNA is unzipped, and then what happens is for every DNA nucleotide that's there, the matching base attaches to it with a sugar and a phosphate. So here the G is there from the original DNA, and then here a cytosine comes in with a sugar and a phosphate to attach to it. Here the original DNA has a thymine, so a new adenine with a sugar and a phosphate comes in. Same thing here, a thymine, a new adenine comes in to attach to it. And that happens to the other side as well. And then at the end, you're going to get two copies of the DNA, and they're going to be exact copies of each other. Here's a little animation of how it looks like. Remember here, the circles represent the phosphates, and then these pentagons are the sugars, and right in the middle is where you can see the bases. Okay, so it's going to zoom out so that you can see the two strands, and it's twisted. So it looks like a ladder, but it's twisted. So right before the DNA has to be copied, it has to be unzipped. So there's a special molecule that unzips the two, and these two strands, we call them the parent strand. And if you see, the new bases along with the sugars and the phosphates will be attached on each side of the parent strands, and it's going to continue until it's done and you have two strands of the DNA. So to continue with a little bit of a review, we already know that DNA is really important. It gives us instructions for how our cells work, and also instructions for building proteins, and those proteins are important for helping us to make traits. So for instance, for your hair, there's proteins that make up the hair color, there's proteins that make up your skin color, and things like that. Now, proteins are a macromolecule. Remember, macro means big, so it's a big molecule made up of smaller pieces, and those smaller pieces are called amino acids. So think of this as one big protein, and these individual circles are amino acids. Now, we have to put all these amino acids together to make that protein. To continue with our review, DNA, we can see it in the form of a chromosome. Now, remember, humans have 46 chromosomes. And all that a chromosome is just DNA, and it's wrapped into this X-like structure. And let's say if we zoomed in on this little part of the chromosome, we can see the DNA. And here it's sectioned off into different genes. So remember, a gene is just a portion of the DNA that gives instructions for a protein that will help us with a trait. So maybe this gene here will give instructions for making a protein that helps you determine your skin color. Maybe gene 2 is going to give instructions for making a protein to determine your eye color. Now, if we take this gene and then we made it bigger, we can see all the nucleotides. So here we have the phosphate, the sugar, and a base. Phosphate, sugar, and a base. And we see it's kind of like a ladder. Now, the way that your body reads your DNA to make proteins is it sections off the DNA in groups of 3, and we call them codons. So here we have one nucleotide, a phosphate, sugar, and a base. It's attached to a second nucleotide and a third nucleotide, and that's how our body reads your DNA, always in groups of 3, which we call a codon, before it moves to the next section, the next codon. So how does our body do that? How does it take the DNA, read the instructions in the DNA, and then make proteins? Well, I'm going to explain it in the same way that we make buildings. Before we construct a building, let's say like our school, there had to have been a master plan. And a master plan is basically just a drawing and has all the instructions of where the classrooms are going to be, the bathrooms, how many levels, where the auditorium is going to be. It has all the instructions for building that school. Now, this one and only master plan, we can't just have one copy of it because it has to get to a lot of different people who would maybe want to check it. Maybe there's needs to be some changes. And also, if something happens to our master plan, maybe it gets lost, maybe it can get ruined, we need to make copies of it just in case something happens to the master plan. And we call those copied versions of the master plan, we call them blueprints. And it's just basically photocopies, and that's why this is here. It's just multiple copies of the original plan. Now, those blueprints, once they're approved, they're eventually going to have to go to the construction workers because they're the ones that are going to read the plans and make the school. So how is that like what happens in our body with taking the DNA and making proteins? Well, the master plan, those instructions for making the school, is like the DNA. Now, the DNA, remember, it's inside of the nucleus. So the DNA, it has the instructions for making the protein. Now, how do we get the DNA that's in the nucleus to make the protein? Well, before we do that, we got to make copies. And like we said, the copies of the master plan are called blueprints. We got to make a copy of the DNA, and we call it RNA. And we make a copy of it so that we can send it out of the nucleus so that we can send it to the ribosome, because the ribosome will read the instructions and will make the protein. Now, I want you to come up with your own example using your CTE. So if you are in construction, I don't want you to have the same exact example. I want you to have an example. What would your analogy be for DNA? Some type of instruction. Then we have to make a copy of it. What would that copy be? Would it be a photocopy? Would it be typed out? Would it be a message, an email? Who is that instruction going to go to, which in this case would be the ribosome? And when they read the instructions, what are they going to make? What are they going to build? What are they going to complete? So whether you're in cosmetology or culinary or business or web design, whatever it is, I need you to come up with an analogy for these 4 parts. What would be the DNA, the instructions? What would be the copied version of it? Or how would you copy it? Who would it go to? And that's the ribosome. And what would they make with those instructions? So I'm going to give you another example just so that it can help you understand this process of how we take the DNA and make proteins out of those instructions. Let's say the end result here, what we're going to make are cookies. And let's say you have a recipe for making those cookies, and that recipe will represent the DNA. So let's say your friend wants to make those cookies, but you don't want to give your one and only copy of your recipe or the DNA to your friend because something might happen to it by accident. So instead, you're going to send your friend, let's say, a text message with the recipe. So that text message represents the RNA, the copied version of the instructions. Now that text message, who is it going to go to? It's going to go to your friend, and your friend is going to represent the ribosome. Now, what is your friend going to do with that text message? Your friend is going to use it to make the cookies, or in this case, at the end, what's made is a protein. So this whole process is how we make proteins in our bodies, but we're going to focus on this first part here. How do we get the DNA and make a copy of it first? And that is called DNA transcription. Now, in order to understand this first part, how do we get the DNA and copy it, we got to understand what RNA is. And RNA, just like DNA, is a nucleic acid, and it's made up of smaller pieces called nucleotides. Now here is a picture of an RNA, and you should be able to see some differences between RNA and DNA. The first thing is RNA has the sugar ribose instead of deoxyribose. That's why RNA is spelled that way, because the R stands for ribo and then the Na stands for nucleic acid. So ribonucleic acid. And if you see, all of the blue pieces here are the sugar, the ribose. The other thing that's different is RNA is only one strand, not two, like DNA. And then the last thing is these bases. So just like DNA, RNA has the base adenine, guanine, and cytosine. But the only difference is RNA has the base uracil instead of thymine. Now remember when we were taking a look at matching DNA bases together? Okay, so remember whenever we see a G for DNA, it matches with a C for DNA. Same thing with the C for DNA, we'll match it with a G. And if we have a T for DNA, it matches with an A, and an A for DNA, it matches with a T. Now the thing is, when we copy DNA and turn it into RNA, we got to match up the bases. So whenever we see a G for DNA, it's going to do the same thing. It's going to match it up with a C for RNA. When we see a C for DNA, it's going to do the same thing and match it with a G for RNA. How about for a T for DNA? It's going to match it for an A for RNA. Now here's the difference. Whenever we see an A for DNA, instead of having thymine, it has U for uracil. So now that we know more about RNA, we're going to take a look at the first part, which is transcription. And all we're doing here is we're copying DNA and turning it into RNA. And we're not making a protein yet. So how does this happen? Now remember that the DNA has two strands. So we have to unzip it. So there's going to be an enzyme, which is just a molecule that it's called helicase, and it's going to unzip the two strands at places called promoters. We don't want to do the whole entire DNA. We just want to copy the certain part of the DNA that we need instructions for. So it's going to unzip. Then here we see the blue pieces are the DNA and they're unzipped. And what's going to happen is we're going to have another molecule called RNA polymerase. And every time it says a DNA, we're going to match an RNA to it to make the copy. So for instance, here is thymine for DNA. We're going to attach an adenine, an A to it, along with a sugar and a phosphate. Here we have adenine, but remember, now it's going to be a U for RNA with a sugar and a phosphate. Here's G for DNA, and we're going to match it with a C for RNA. And it's going to go ahead and keep matching an RNA to a DNA. And this red piece here, once it's done matching, it's called an RNA. It's called specifically a messenger RNA. You can think it carries the message of the DNA. And once that's done, the RNA is going to leave and go to the ribosome. So here's a quick animation of how that all looks like. So this is the DNA and it's at inside the nucleus, and we have to unzip only a portion of it. We don't want to unzip the whole DNA. We only want to unzip the part that we need the instructions for. And there's that molecule, helicase, and it's unzipping the section that we need to copy. And then we're going to get another molecule, which is the purple structure. It's called RNA polymerase. And every time it sees a DNA, it's going to match it with an RNA. And once it's done copying that section, the DNA is going to zip back up, and what is starting to move is the messenger RNA. And now the messenger RNA is going to leave the nucleus, and it's going to head over to the ribosome so that the ribosome can read these instructions in this messenger RNA and make a protein out of it. So this section of copying the DNA and turning it into messenger RNA is called transcription. So let's do some examples. In this first part, I want you to copy the DNA top strand, that just means one part of the DNA, and then match it with its complementary DNA bases. Now remember, G always matches with C and vice versa, and T matches with A and vice versa. So here, this first codon, and remember, a codon is just every group of 3 nucleotides. Okay, and this is how our body reads the DNA. So the first nitrogen base in this first codon, T, matches with A. Then G matches with C, and then C matches with G. So the complementary or matching DNA bases would be ACG. The next one, A matches with T, G matches with C. So the next codon would be TCC. And this is what the rest of the DNA would look like. Now let's transcribe by taking one of these DNA strands and then making it into a messenger RNA. So this first codon for DNA, TGC, when we match it to the RNA bases, T for DNA matches with A for RNA, G for DNA matches with C for RNA, and then C for DNA matches with G for RNA. So that first codon for RNA is ACG. The next one, for DNA, it's AGG. So A for DNA, now it's different. It matches with U for RNA. And then G for DNA matches with C for RNA. So the next codon for RNA would be UCC. What about the next one? AAT. A for DNA matches with U for RNA. And then T for DNA matches with A. So it should look like UUA. And here are the last 2 codons. Now let's try another example here. Again, I want you to take this top DNA strand, just one of the sides of the DNA, and match the bases to its complementary DNA bases. So remember, here's DNA ACA, and if we were to match it to its bases, it would look like this, TGT. Here, TCT matches with the bases AGA. And here are the rest of the DNA bases that match up with the top strand. Now let's transcribe it. That means we're going to take only one of the DNA strands and we're going to copy it and change it into RNA. So remember, ACA, A for DNA matches with U, C matches with G. So it would be UGU for RNA. Then TCT, remember T for DNA matches with A and C matches with G for RNA. And these are the RNA codons at the end. And like I said, this messenger RNA is what's going to be read by the ribosome so it can make whatever protein that our bodies need.